This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 189, recorded on November 8th, 2018. I'm Vincent Racaniello, and you are listening to the podcast that explores unseen life on Earth. Joining me today, from Small Things Considered, Elio Schaechter.
Hello there. How are you?
I'm well. How are you?
Middling well.
Good to hear you again. Also joining us from Ann Arbor, Michigan, Michele Swanson.
Hello.
Welcome back. We missed you for one podcast there. And from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
People complain on TWiM that I spend too long introducing people, so I just made it quick. See? I can do it.
Oh, no.
People can complain about anything, right?
They try.
They mostly
That's a democracy.
On TWiM, they mostly complain about the weather. But I always say you can't do anything about it, so why complain? That's funny, isn't it? All right.
Terrific.
Before we go do some science here, I have a little request for all of our listeners. Some of you may know, or if you don't, you're going to learn that we do have a YouTube channel, youtube.com/PROFVRR. youtube.com/ProfVRR. That's me, Professor VRR. It's my YouTube channel where I have videos of my lectures, select TWiM, TWIP episodes, short videos, and other things. And I learned recently that YouTube is giving out educational grants for educational content. And I would like to apply for one. But you need 25,000 subscribers. And at the moment, I've made a plea on all the possible places I can. At the moment, we have 16,600 subscribers. So
that's terrific.
You have an additional 42. You're up to 16,642.
Right. So subscribe if you haven't already. It's free.
Is it clear how to subscribe?
Yeah. If you just go to youtube.com/PROFVRR, there's a big red subscribe button right there in the front page. I'll tell you exactly where it is. It is located
in the upper right-hand corner.
Upper right-hand. I don't see it because that's my channel, so I'm not going to see you subscribe. Have you subscribed, Michael?
I have indeed. Good. And if you click the bell next to it, what will happen is anytime Vincent posts new content, whether it be from TWiM, TWIP, whatever podcast he has, when you click that little bell, anytime there's a new podcast posted, it'll show up on Vincent's channel, and you will then be able to know that there's something new for you to listen to.
That's right. So do that, and if we get to 25,000, I will apply for support.
And not only do that, but tell all the people in your group, your fellow students, lab mates, friends, and neighbors to also do the same.
Yes. Needs to go viral.
Please. Yeah.
All right. So thanks for your support. All right. Today we have something a little different from Elio.
Yes. Let me tell you what's different about it. I'm going to talk about a report that was put out by the Kew Royal Botanical Gardens, a very important institution in England, about the state of the world's fungi 2018. So instead of talking about an article, which we normally do, this time I'm going to give you a small summary of what this report says. And see if you find it interesting, because fungi, by jolly, are mighty important. But we're going to play a little game here. In addition to listening, those of you who are not driving or performing an experiment may want to keep a tally. As I mentioned the item, check off, yes, I knew this, or no, I didn't. And then let us know what you think. If you think that you are up on the state of the world's fungi, so much the better. If you learn something from this, even better. Okay?
Can we play along? Can I play along?
Yes, you can.
All right.
There's going to be a test later, Michele.
Just for us. Okay. By coincidence, in today's Small Things Considered, now today is not the day when TWiM appears, but it will in time. Anyhow, in today's Small Things Considered, Roberto Colter, co-blogger, wrote a piece about just that. So he wrote a very lovely piece. Okay. So this is a list. It's a laundry list, if you wish, but written extremely well, with a great deal of thought, beautiful pictures. It's worth going to. It's called The State of the World's Fungi 2018. If you Google that, you'll get to the Kew Royal Botanical Gardens site. Okay, let me summarize first what they talk about. They talk about new species being discovered, about the usefulness of fungi in food production and bioremediation. By the way, you don't have to check this off because this is just general. This is just the summary of what is in the article. Classifying fungi through large-scale genome sequencing, the importance of fungal-plant interactions, both in promoting plant health and causing plant disease, the effects of climate change on fungal ecology, catalog of the current state of the world's fungi, and conservation efforts aimed at protecting endangered species. So that's what this is about. Now, I'm not going to present it in the same order just because. So the first thing I want to tell you about is that in research, fungi appear very, very prominent. You know that yeast is the E. coli of the eukaryotic world. It's the most studied organism other than humans, and a tremendous amount is known about it. In fact, since 1910, a quarter of the Nobel Prizes in medicine were awarded for work based on yeast. So that tells you. And yeast is, of course, kind of a little substitute for humans because 7% of yeast genes can be replaced by human genes. So, it's a great organism. Okay, so with that in mind, let's go over a list of items. Let's start with recreational uses, okay? About 200 species of fungi are thought to be hallucinogenic. Okay? We don't know the real number, but that's about right. And Mexico has the highest recorded number. They have 76 species, of which 46 are in the genus Psilocybe. Now, I'm going to add something that's not quite in the report. Psilocybin, which is the active principle of the magic mushrooms, is now increasingly popular in research, in medical research, and it's used for treatment of drug addiction, depression, pain. It seems to have a wonderful effect, and there's efforts afoot to remove it from the totally forbidden class of drugs to the ones that can be used under supervision, etc. So a lot of research is going to go into it. Anyhow, let's go on to...
So wait, can we anticipate then medical mushrooms?
Yes. No, right. Medical psilocybin.
Okay.
Pill. Now, the reason for not giving you a mushroom is because although you may think you've identified, you may misidentify it, and it may have something else in it. So it's not a good idea. And in fact, I would say, please, if you're interested in recreational uses, try to really know what you're doing. The best thing to do would be, in fact, to get hold of the chemical and use that, because swallowing mushrooms indiscriminately is a good path to a trip to the emergency room. So
or the coroner.
That's right.
Or a trip.
Or a trip, right? A final trip. So be careful with this, okay? Anyhow, we can go on with that, but let me go and take on the subject of food. Mushrooms are, of course, the best expression of food produced by a fungus. The global market of edible mushroom is skyrocketing. It is now $42 billion in the U.S. per year. About 350 species are routinely collected and eaten. There's a big difference between cultivation and eating, because the number of cultivated mushrooms in the supermarket today is really also skyrocketing. In my local regular supermarket, there are 5 different kinds of mushrooms, and they are white buttons, cremini, and portobellos. Now, don't say I never touch anything. Cremini and portobello, the same thing as the white button mushroom, only they've been allowed to grow further. So remember that. Same species, Agaricus bisporus. Shiitake, which very, very common now. oyster mushrooms, of which there are many varieties that are interesting. There's one called the giant trumpet. See if you can find it in your supermarket. It's very cheap to grow, very easy to grow, and it's quite delicious. And then there is wood ears, which are mainly for consistency, so the chewy. And gnocchis. Gnocchis are those little things that look like spaghetti with a cap.
Mm-hmm.
Right? They're quite good. Okay.
Elio, which is the one my grandmother used to cut up?
White buttons.
Really?
Yeah.
So they can be quite large, right?
Sure.
Okay.
They can be quite large. If they let them grow far enough, they make spores, which is on the you see them on the underneath, on the gills underneath the cap, and that's what makes it cremini. Mm-hmm. And if you wait long enough, it makes it portobellos.
Portobello, right? Love them.
Portobello, beautiful port. Anyhow, in food, we should also mention, of course, that brewers and bakers' yeast underpin the making of breads and essentially all, if not most, alcoholic drink productions. So, so much for fungi being our friends. They also participate in ripening of cheese. Many cheeses are ripened by that. In addition, they're used in making miso and tempeh, a cousin of it, which is soy material which is solidified. It's done by fermentation with a mold. Molds make food coloring, soy sauce, etc. So great uses of fungi in foods. Industrial products. Fungi contribute to the production of a large number of essential compounds like antibiotics, statins. I don't know if there's statins to combat high cholesterol. I don't know if they are essential, but they're important. And compounds like cyclosporin used in tissue, in organ transplant to lower the immune response. They also are used, and this is less known, in modifying steroids chemically. They may not make the steroid backbone, but they contribute to the modification. To give you an idea, in vaccines and therapeutic proteins, about 15 to 20% of all are made in yeast. And then molds are used for making vitamin B12 and a whole lot of enzymes. Before I go into enzymes, though, I found a cute one. There is a platform chemical. A platform chemical is a chemical that's used to make plastics. So in various ways. It's called itaconic acid. And guess where you find it? You find it in Lego toys.
Great.
So fungi are responsible for Legos?
Indeed.
That is something that's going on the list. That's something I did not know.
Okay, very good. Okay, in addition, the mycelia, the filaments that fungi made, molds and mushrooms alike, a hair-like network of cells. They can be grown in labs to make lab-grown meat. Look out for that, because apparently it's very, very good, and it's a substitute for meat. It doesn't look like a chop, but it can be made to look like anything, and it can be very good.
So it has the chewy texture that people are used to with meat?
I guess. Yeah.
Interesting.
Well, it probably depends on the binder you put in. You have to put in some binders, and I imagine you can tamper with that. But look out for non-cow-made meat made by fungi. In addition, they can be used to make the scaffolds for 3D organ printing. That is, if you want to make an artificial organ, you need to put the cells on something, and the scaffolds can be made out of the mycelium of fungi. And there's a huge number of uses like that. They are used, people are talking about using them to making biofabricated leather. Imagine that.
Wow.
So anyhow, enzymes. There's a whole lot of enzymes which are made by molds. One of them that you may not have heard of, but you know the consequence of it. When you drink soft drinks which are artificially sweetened, you're likely to imbibe high fructose corn syrup. Well, what's that? That's corn, starch from corn, which has been modified into fructose by an enzyme called glucoamylase of a species of Aspergillus. I mention Aspergillus because it's a species of mold which is extraordinarily common, perhaps the most common mold in our environment, and it's used in a tremendous number of different industrial productions. One use that you may not have heard of is that certain cellulases from a thermophilic fungus, so this is good because you can use the enzyme at high temperatures, are used to trim the fine cotton threads on the surface of cotton fabric. And this makes for a smoother feel of the cotton.
Like
That's
acid wash jeans or stone wash jeans?
No, I don't think. Maybe. I wouldn't know, but they can be these threads apparently interfere with the fine feel of cotton, and they can be removed with these enzymes. They, of course, enzymes from yeast, from molds, are used to remove stains attached to fabric surfaces, and that's very helpful because it allows you to wash your clothes at a lower temperature, which is very good for the environment. And they're added to detergents. They also can be used are used to turn crop waste into bioethanol. This is important because if you want to make ethanol out of corn, use the corn ears, and the rest you throw away. But the rest is cellulose, which is perfectly good carbohydrate, which can be broken down by enzymes made by fungi into something which can be fermented into ethanol, etc., etc. So there's lots more of that. I don't think I want to go into much more on that. In agriculture and in the environment, there is fungi have a tremendous importance. The main one, which is not emphasized in this article as much as I would have liked to see, it's mentioned but not highlighted as much, is that fungi are the great recyclers. If it weren't for fungi, trees that decay in the wood would not decay. Trees which are dead would remain dead. And if it weren't for the fungi, if you wanted to go for a walk in the wood, you'd have to take a chainsaw along because you could make your way.
Wow.
So obviously this means that they participate in a major way in the cycle of carbon in nature. They do release carbon in the form of CO2 by the decomposition of plants. That sounds like a thing which is not desirable if you think that too much carbon is a bad idea, but we'll see to it that they make up for it by allowing plants to grow better and therefore absorb CO2 or use CO2 for photosynthesis. So it's a mixed bag. They increase the amount of carbon dioxide and they decrease it at the same time. One item that I didn't know about is that enzymes from a mold are added to animal feed and they increase the weight of cattle by up to 13%. I didn't know that. So they say food supplement. Now, the big thing, of course, is that fungi make a relationship with the roots of trees, the mycorrhizal relationship. 90% of all plants have mycorrhizal relationship. And this is done by only about 2% of all fungal species. So these are specialized fungi that undertake mycorrhizal relationship. Just to remind you, they provide the plant with water and minerals, especially phosphorus, from the soil. So they're like drinking straws because they extend the reach of a tree by a huge factor. It can be, in fact, the size of a county. And if you wish, it can be even thought of as the size of the whole landmass because mycorrhizal fungi make anastomosis with other species. So in return for that, they get the product of photosynthesis. So the plants receive water and nutrients, and the fungi receive sugars. In addition, they increase resistance to pests and disease. Mycorrhizal fungi and endophytes, that is fungi which grow inside of tissue, in some cases allow plants to withstand dryness. And they also, as I said, they will protect plants from certain parasites, so they're kind of like bodyguards, not just the drinking straws for the tree, but also bodyguards. So by increasing the growth of trees, of course, they decrease the amount of carbon dioxide in the atmosphere. Were you going to say something, Michele?
No.
Okay.
I'm just being humbled by the number of no's I've got.
Yes. Yeah.
So fungi degrade pollutants like pesticides, dyes, explosives, and accumulate heavy metals, so they can even be thought to be able to clean up toxic waste contaminated with radioactive material or polychlorinated biphenyl chemicals. So they're useful in that way. And
Phenorichides species, the Phenorichides are really especially good at that because they effectively make these molecular grenades, and the explosive in the grenade is, if you will, radical oxygen. And as the Phenorichidae throw out these radical oxygen grenades, they can actually bust up lignin. And lignin, of course, is what holds the tree vertical. And so it's one of the most refractory natural chemicals on our planet. And the fungi just liquefy it because they throw these tremendous grenades at this very, to use Dixon's phrase, chicken wire chemistry. And it's just remarkable.
Yeah, that's right. So they work as in that sense. They also do something which you may not know. They change the structure of soil by formation of soil aggregates and pores, which enhances the growth of plants. So they are really the tillers of the land as well as everything else. All those are good things. How about bad things? Well, there are plenty of them. Fungal diseases are a worldwide threat to ecosystems. They are things which, I'll give you an example. Until 2010, the disease myrtle rust was known as a problem in plantations of plants introduced into South America. However, in 1910, it was detected in Australia, where it has the potential of infecting 1,000 different native species, including eucalyptus and the paperback tree, Melaleuca. And of course, the number of plant diseases which are caused by fungi is enormous. I just rattle a few names: Dutch elm disease, ash, dieback, dogwood, anthracnose, I never heard of that one, beech bark disease, white pine, blister rust. So are these threats? Yeah.
If you're looking at this report online, they have interactive maps showing specifically the ash dieback in Europe, and it's really quite remarkable. You click on 1992, and you see most of Europe is green. There's just one country with orange, which is indicating that the ash dieback has actually taken place. And as you walk through 1992 to present day, which was 2017, you're going to be shocked the amount of dieback that has occurred across Europe just from looking at this interactive map. They have done a tremendous job with this website, and you can also download each of these as individual chapters if you want to put it on your tablet as you're riding the subway to work, so you have something to read, so you can actually read it if you lose your connection going underground. It's really quite fascinating.
It is
of how they've laid this out and made it so approachable, so you can digest it with time.
Yes. Now the question is, are fungal threat emerging? And the answer is that global warming may have a big impact on this, because climate change opens new regions for fungal pathogens. One study, they say, indicates that fungi are moving towards the poles, following their plant host at the rate of 6 to 7 kilometers a year as the earth warms. And of course, the planting of monocultures, which is increasing. Farmers, industrial farmers, increasingly use one species of potatoes, one species of green beans, and that, of course, makes them more susceptible to disease. So disease in plants, not so much in animals, although in animals it's certainly important as well. In immune-compromised people, fungi are the main threat. But in plants, it's bigger and it is important. So that's more or less my take. But let me read you the conclusions so that you get the feel for their voice rather than mine. They say, despite early recognition of the importance of fungi for human well-being and archaeological evidence for human uses of fungi in food, drinks, and medicines going back at least 6,000 years, historically, they have remained in the shadow when compared with research on plants and animals and bacteria. In fact, many of the early writings on fungi assumed they were simple or lower plants. It was not until detailed work on fungal features, including the cell wall. But incidentally, the cell wall is contains chitin, just like the skeletons of the exoskeleton of insects. Anyhow, and it wasn't until works on fungi features, including cell walls, methods for digesting and storing food, and DNA, it became apparent that, in fact, a kingdom of their own right, they belong to a kingdom of their own right, closer to animals than plants. For example, it says here, most fungi have cell walls composed primarily of chitin, a substance that is also found in the exoskeleton of insects and the shells of crabs and lobsters. The realization that fungi are close to animals than plants is, however, only one of the remarkable facts to emerge in the past decades. It is now becoming apparent that these organisms, which often cannot see with the naked eye and spend the vast part of their life cycle underground or inside plants and animals, are responsible for incredibly important processes. These include global cycling of nutrients, carbon sequestration, and prevention of desertification in some drought-prone regions of the world. Fungi underpin products and processes that we heavily rely in aspects of everyday life, from critical drugs like statins to synthesis of biofuel, cleaning of environment through bioremediation. Some, like species of Aspergillus, have multiple uses, as diverse as making antibiotics, the synthesis of third-generation contraceptive pills, and cheese production. The global market in edible mushrooms is also huge and increasing. That's pretty much it. So you're going to get a lot of pleasure by reading this because it's extremely well written. Like Michael pointed out, the graphics are amazing, and it's just a pleasure to the mind and a pleasure to the eye.
So there's 10 chapters, folks. And so this will keep you busy for a while, and they're absolutely fascinating.
Yeah.
The website's gorgeous also.
Oh,
it is.
It's so beautiful.
It's like National Geographic except for fungi.
Right.
Yeah.
And so well organized. If you're interested in reading about useful fungi, you click on that, and then you get another beautiful graphic.
That's right.
That lists about 15 different products. You can click on each of those, like washing powders or leather processing, and learn more about that. So you need not feel like you have to go from page 1 to 200 or whatever. You can really go in and follow your interests. It's beautifully done. What a great public service.
Mm-hmm.
Indeed, I think that's a good way to put it. And I hope the listeners will understand why we departed from our usual format in order to highlight this rather remarkable piece of writing.
I have to say, I probably knew 10% of what you said. Right, most of it was new to me.
That much?
Yeah, maybe less.
I didn't appreciate that the Kew Botanical Gardens had such a large collection. They on their website, they say that they have more than 1 million different specimens of fungi at the in their collection, and that collection has been visited by the likes of Charles Darwin and Beatrice Potter.
Yeah, Beatrice Potter was a mycologist on the side. She illustrated mushrooms beautifully. She was a very good painter. Anyhow, the Kew Botanical Garden is indeed one of the world's center for mycology studies. You're very right. Yes. And the other one is at Harvard, the Farlow Library and Herbarium. I
would guess that most people don't know much about fungi except mushrooms, right, from what they eat. So this is great. It's going to teach a lot of people. So.
And they have a chapter on edible mushrooms, too.
Yeah.
Absolutely.
And another chapter on the many contributions that the Chinese have made to this field, because there's a long history of Chinese using fungi in medicine, in food, and other applications. So they get a special highlight here as well.
Great. Well, folks, dive in, stateoftheworldsfungi.org. It's free. Go look at it after you subscribe to my YouTube channel.
Shameless plug.
Thank you, Elio.
My pleasure. I really enjoyed this.
And of course, you're also an aficionado of fungi, right?
I am indeed. I once wrote a book.
That's right.
In the Company of Mushrooms.
That's right. It's still on Amazon. You could buy it. Yeah.
Well, it's.
I think we need to link to that.
Well, you think it's out of press?
It's out of press, but they have used copies. So I don't get any royalties, so that's fine.
Yeah, you can find lots of them in the company. Yeah, there's plenty of used ones always. That's the beauty.
It sold fairly well when it came out, so it's not surprising that there are used copies.
Yeah, all right. Beautiful. Thank you, Elio.
My pleasure.
In an effort to make ASM's podcasts the best they can be, This Week in Microbiology wants to learn more about you, the listeners of TWiM. Please take 5 minutes to answer this short 12-question survey. Surveys help us collect the data needed to seek sponsorship, which would allow us to grow the podcast. You can find the survey at asm.org/TWIMpoll. That's asm.org/TWIMPOLL. Thanks for your time and listening, subscribing, and helping us improve the show. We appreciate the support. All right, on to a paper, and from this, we will hear about this from Michael.
Something completely different. The paper that we're going to talk about today is Regulatory Evolution Drives Evasion of Host Inflammasomes by Salmonella typhimurium. This paper appeared on the 23rd of October, 2018, and it's an open access contribution that appeared in Cell Reports. So the story in brief.
Explain what that means for a second. Not every listener may know what open access means.
Open access means that it's available for anyone listening for public download. So the authors or their grants have paid the publisher all the costs associated with bringing the paper to the journal. And there are real costs associated with publishing a paper. And they have opted to pay that fee so that anyone who wishes to read their manuscript may do so. So it's available. All you need is an internet connection. You can get into it and download it, and it'll be in the show notes so that you can see the beautiful figures that they have in this particular paper. And the paper was authored by Ilias, Mulder, Little, Alanaway, Banda, Perez Morales, Tassi, Chow, Bustamante, Coombs, who are from two institutions. The first group is from the Michael DeGroote Institute for Infectious Disease Research at McMaster University in Hamilton, Ontario, which is in Canada. And the others are from the Departamento de Microbiologica, Molecular Institute de Biotecnologica at the Universidad Nacional Autonomía de Mexico.
Elio's laughing.
You're getting half the world is smiling right now.
Yeah, well, it's the problem with my Chicago accent. From Cuernavaca, Morales, Mexico. So it's a fascinating story. And the story in brief is bacterial pathogens tune their gene expression based on the niche or the location they find themselves in. The story today is going to describe the identification of a very clever immune evasion strategy that evolved in Salmonella enterica serovar typhimurium, which I'm just going to refer to now forevermore as Salmonella typhimurium or just typhimurium, that couples repression of its flagellar motility once inside the host. Recall that strains of Salmonella that are infectious in humans often become facultative intracellular parasites. And so intuitively, it makes sense that they turn off motility because once you're inside, where do you need to go? And we're going to describe how these authors worked through this remarkable observation that once inside the cell, by turning off motility, they effectively lie to the eukaryotic cell so that it doesn't turn on all the immune activation parts that Michele studies in her lab. Don't you work on inflammasomes as well, Michele, with Legionella?
Yeah, that's right. I won't take the punchline, but we made similar observations in Legionella.
Mm-hmm. So the story has four parts.
It's interesting.
The story has four parts. First, we're going to learn about the horizontal acquisition of the regulatory element, SSRB, which is nothing more than a transcriptional factor that finds the right piece of DNA, activates it, or turns it off for transcription. And this is part of a two-component regulatory system that many of you are familiar with. And the second piece is they begin to dissect how this regulatory molecule divergently regulates flagellar motility in the Typhimurium versus the ancestral strain Bongori. Salmonella Bongori is presumably one of the ancestral strains of the Salmonella that become facultative intracellular parasites of mammals.
Isn't that a rock star, Bongori?
I think it's BonJovi, and he's from Jersey, so you'll probably get notes from him. The third piece of the story is they were able to beautifully show an evolved SSB binding region. So this is where the transcriptional factor binds in the nucleic acid upstream of the flagellar operon that then drives the repression of Typhimurium once it's inside the cell. And then finally, the fourth piece of their story is that this motility repression then physically promotes the evasion of the host inflammasome activation during the infectious process once the salmonella is inside the cell. And if you studied any immunology, you know that once one of our immune cells effectively has engulfed bacterium, it turns on a whole suite of genes, and the net consequence is it gets rid of the bacterium. But it does this through this beautiful cascade of immune regulatory molecules, and that's effectively how it clears the infection. The hallmark of a salmonella infection is they live happily ever after inside the immune cells, and this is the story of how they're able to do this. So now, as background, if you're unfamiliar with salmonella, is you need to know that the evolution of salmonella as a pathogen has been shaped prominently by genomic evolution via horizontal gene transfer. Now, the horizontal gene transfer effectively was accomplished by the acquisition of pathogenicity islands that we've talked about in the past. And these pathogenicity islands are differentially distributed in different strains in serovars. And if you have trouble sleeping, you may want to learn about the taxonomy of salmonella because it's long and complicated. And I'll put a specific short piece in the show notes about the CDC talking about how the naming conventions of salmonella have evolved with time. But the authors of this manuscript tell us that the acquisition of the pathogenicity island number 2 in salmonella followed the divergence of Salmonella typhimurium from Salmonella bongori. Elio asked me a question via email before this paper. Why Bongori? Well, Bongori simply because that is, if you will, the evolutionary great-great-great-grandfather of typhimurium.
Today, it affects mainly reptiles and invertebrates, right?
Mm-hmm.
Not invertebrates, non-mammals.
Yeah, and the principal difference is Typhimurium has the ability to survive and replicate within immune cells, whereas BonJovi does not. And so the principal difference is, one, the Typhimurium is capable of being a facultative intracellular parasite, while BonJovi is not. So this then, acquisition of this pathogenicity island enables Salmonella, by virtue of the fact of acquiring this pathogenicity island, to exploit this new intracellular niche, because nothing's going to attack it once it's inside the attacking cell of our system. Second, when Salmonella typhimurium acquired the pathogenicity island, it got for free this two-component regulatory system that the authors are investigating. And if you recall that two-component regulatory systems, the hallmark of this canonical system is one molecule adds a high-energy phosphate to the other molecule, and when that molecule acquires the high-energy phosphate or loses the high-energy phosphate, it effectively acquires the ability to modify transcription. It either modifies it up, or in some cases, it can modify it down, depending on what its specific function is going to be. So we're going to talk principally about the B component of this two-component regulatory system, and it tells the cell, if you will, when it came in from the cold. So that's what this regulatory system that was on the type, which was on the pathogenicity island, does. It effectively says, okay, you're now inside, you can do other things. One of the curious things about this regulatory system is that under acidic conditions, the sensor kinase adds the high-energy phosphate to the SSRB response regulator, if you will, activating it so that it can now bind to the proper regulatory regions of the target genes to regulate their expression directly or through counter-silencing of protein systems called HNSs. And as an aside, I don't believe that we've ever talked about these nucleoid-associated protein, which is, just has the abbreviation of H-NS, which is able to recognize and bind AT-rich, horizontally acquired genes, which are typically the hallmark of pathogenicity islands. Pathogenicity islands in general have different G+C ratios than their host, and often they're more AT-rich. And so
that's how they were originally
that's how
found,
they were discovered. Right. And this HNS system, and after I read this, I said we probably should look for a paper that talks about these nucleoid-associated proteins that can silence expression. Now,
but in general, it's a way for the bacterium to protect itself from foreign DNA.
Correct.
To shut it off.
Correct.
Yeah. Wrap it up, shut it off.
Wrap it up.
For the non-microbiologists, these proteins vaguely act in something like histones in eukaryotic cells.
Yes. And it's sort of the, if you will, the histone analog.
Right.
So the take home then is that collectively the genes turned on by the activated transcription factor SSRB by the acidic conditions contribute then to the overall collective fitness of the typhimurium once inside the host. The third bit of background we need to know before we move deep into the deep end of the paper is that many of the genes outside of the pathogenicity island, so this is like genes in the normal chromosome that are controlled by the same transcription factor that is turning things on in the pathogenicity island or turning things off in the pathogenicity island, have equivalent genes in the evolutionary ancestor that they're studying, which is the Salmonella BonJovi. And recall that the principal difference between BonJovi and the typhimurium is the absence of this pathogenicity island. So our journey is going to have 4 steps. The first 2 portions of our story concern comparative transcriptomics and what they learn. The second piece of our journey specifically looks at the functional classes of the genes that are turned on or turned off in Typhimurium and Bongori. And then they ultimately demonstrate that this transcription system mediates the repression of the flagellar base motility, serving to limit the inflammasome activation during intracellular infection of macrophages. And they actually do the full-scale animal activity. So the first stop on our journey.
All this in 10 minutes, right?
I'm going to try. So they do the traditional things. They isolate total RNA, but the clever thing they needed to do is rather than worrying about SSRA and SSRB, they just cloned SSRB and they put it into a strain of Salmonella typhimurium and also into a strain of Bongori. So they have this active transcription factor that's consistently turned on, and then they, of course, have the isogenic control that's lacking it. So they isolate total RNA from vector and the strains that they have modified, and they identified about 331 genes that are regulated by this molecule, which is about 6% of the genome of Typhimurium, and Typhimurium has about 5,500 genes in total. So 331 genes were upregulated, and they tell you what upregulated means, which is operationally transcription is up twofold. And then 224 genes were downregulated. And again, downregulated is greater than twofold down. Of those genes with altered regulation in Salmonella typhimurium, 62% of the upregulated genes and 87% of the downregulated genes had orthologs in Bongori. And this is interesting considering that the B system, this transcription regulatory system, only came to Typhimurium with the pathogenicity. So they were most interested in the orthologic genes that was then going to be controlled by this transcription system. And they were most interested in the ones where there was discordant regulation between the two species, where Typhimurium was up, Bongori had to be down. And so here is where they make their fundamental hypothesis that this was likely an outcome of regulatory evolution, principally driven by fitness. So there were 20 genes that were upregulated in Typhimurium, but downregulated in Bongori, and 14 genes downregulated in Typhimurium, but upregulated in Bongori. As you might guess, they next assign the genes to functional classes. And what they learned is they had to do with motility. So the next section of their paper describes how this transcription complex divergently regulates flagellar motility in Typhimurium and Bongori. Now, here's where you get really impressed with their figures. Their figures are quite striking, and while we know gene expression is but step one in our regulatory story, the proof is often in measuring the activity of the resultant gene product, or here, they're actually looking for motility. And so they show you a motility assay in their figure. And this is the most curious case or curious observation. And here the discordant effect of the transcription factor on motility was confirmed by bacterial swimming assays in which the transcription factor SSRB significantly decreased swimming in Typhimurium, but increased it in Bongori. And when you look at the photograph of the swimming assay in the top portion of the figure, and then they present the control without the SSRB and Typhimurium, it's the swimming is quite rapid, and then with the SSRB, it's very much lower, and it's significant out to 4 stars. So that means 0.001. So it was indeed significant. And then the BonJovi is literally flipped. So the swimming assays correlated beautifully with a significant decrease in the number of flagella present on Typhimurium and a significant increase in the number of flagella on BonJovi. And they actually...
Yeah, and Michael, if I could just interject.
Please do.
I was able to correspond with our first author, Bushra Ilyas, and she said that this was one of the most exciting moments in the project. She was working on this, collecting pellets of bacterial cells to make the RNA and do the large transcriptional profile, but they noticed that in the pellets, they either got a really tight bacterial pellet or a really loose one, and it was attributed to whether or not they expressed the SSRB and whether or not it was Typhimurium or BonJovi. So just by looking at their cell pellets, they knew that they had a distinct difference, and then they confirmed that by doing the molecular analysis that you just described.
That is funny.
Yeah.
I mean, this is so elegant because normally when you think about transcription stories, they go from RNA, they show you a bunch of Western blots with the proteins going up or down. But here we're actually showing motility, and it really makes sense. You don't need to be motile once you're inside a cell. You want to remain it's expensive. And so you can understand the evolution of how this all came about. And so that would then takes us to the next component of their study, is where they begin to understand and look at what this regulatory complex is doing vis-a-vis the flagellar complex. And so they go in. You have to have a little digression to learn about flagellar gene expression in Salmonella, and there's a hierarchy of 3 different promoter classes, followed by the apparatus and chemotaxis genes through class 2 and class 3 promoters. But the bottom line, again, by confirming it via RNA sequencing, they show that the B-dependent repression of all levels of the motility hierarchy in Salmonella typhimurium. They do this beautifully by RT quantitative PCR, and it's, again, a very striking figure. BonJovi is up, Typhimurium is down. It's crystal clear. You just look at their figure, and all the Typhimurium is going negative, all the BonJovi is going positive. They remind us of previous work that has gone on before by Earhart and Dersch, that the flagellar promoter system is a hotspot for transcriptional input into motility regulatory cascade. And it's almost similar to what we recall from the regulatory cascade when you think about sporulation. There's a specific timing to it. And so their next set of experiments specifically look at trying to understand how this transcriptional complex is interacting with the various gene products. And although they found that they looked at one specific regulator, and that's the HIL-D, which is a transcriptional regulator shared by Typhimurium and BonJovi that is known to activate the flagellar gene complex, FLHDC. And although they found that the Purify HIL-D bound equally well to Typhimurium and BonJovi FLHDC promoters, the interaction did not affect the binding of this global regulatory factor, SSRB, to Typhimurium or BonJovi, vice versa, thus ruling out competitive inhibition by this transcriptional activator. And if you know a little bit about transcriptional activators, they often don't work by competitive inhibition. They're typically binding someplace else. And that's effectively where they take us in the next component of the paper, where they beautifully dissect, and they did this by good old gel shift. And that is where you add a protein to some nucleic acid, and when the protein binds, you see a shift in the mobility on a gel based on the binding of the transcriptional regulatory protein onto the nucleic acid. And so they slice and dice without going into detail, giving you the landmarks. You can read that in the paper. And so they were able to dissect precisely where this factor SSRBC was specifically binding. So that effectively they've now identified it. So now it's on to the behavior that this B-dependent repression of flagellar gene expression limits inflammasome activation. So we know,
but it's also worth pointing out from that analysis that it was beautifully aesthetically pleasing that the direct binding of the regulatory protein was at the top of that regulatory cascade for the flagellar operon. It was the master regulator, FLHD. So that shuts off the entire class 1, class 2, class 3 cascade of flagellar genes.
And getting back to Michelle's earlier comment about the expense of expressing motility in the wrong spot, by doing this upstream, you effectively shut down and literally, if you will, save lots of money in terms of energy for the cell. And so then this then takes us to the whole notion of how the underlying hypothesis of the authors was that the selective driver, if you will, behind all of this evolution of this beautiful molecular biology that they've shown us so far was simply to repress motility, which in turn was to limit caspase-1 activation and the release of IL-1 beta. So how did they prove this? Again,
and that, just to bring the listeners up to speed, that is an innate immune barrier. So all macrophages, for example, have that. If they recognize minute quantities of flagella, they turn on this fever-producing and cell death pathway.
And it's quite specific. And those of you who know that flagella, there are more than one flavor often in Salmonella, and they can switch the flagellin protein type that's used to make flagellin, and they do that via a very elegant system of phase variation in Salmonella typhimurium. So what they now have done is now we're going into mice. So we're asking, does this really work in an animal situation? So mice were either infected with typhimurium carrying a strain that didn't produce SSRB, or ones that did, that expressed a luciferase transcriptional fusion that showed that typhimurium with the SSRB positive regulatory element significantly reduced luciferase activity compared with typhimurium lacking this regulatory molecule. They then demonstrated that, similar to their in vitro results, the fly C was more abundant in macrophages infected with typhimurium lacking SSRB and was undetectable in cells infected with SSRB positive constructs within them. And they have the BonJovi controls as well in their figure. And it's really quite elegant to see how well they're able to do it. So we're coming to the last experiments. So you're probably fatigued, but I encourage you to take a look at what they have done, because it's a tremendously elegant story. They next wanted to ask the question, the effect of these fly C levels on inflammasome activation. And this was tested in bone marrow-derived macrophages using secretion of caspase-1 and the pro-inflammatory cytokines IL-1 beta as readouts following infection with either typhimurium and BonJovi. And here again, the SSRB negative typhimurium triggered tremendous amounts of caspase-1 processing, enriching for the active 20 kilodalton subunit, suggesting that the inflammasome is on. and significantly increase the secretion of IL-1 beta. And the ones that had SSRB were off. And BonJovi was flipped, and the activity was dependent on both the Fly-C and SSRB, because deletion of Fly-C and the SSRB minus typhimurium, or expression of the SSRB and the Fly-C positive typhimurium, prevented caspase processing and significantly inhibited IL-1 beta release from the bone marrow infected macrophages. In contrast, the BonJovi was the opposite. And then they did the complementary experiments treating bone marrow macrophages with caspase-1 inhibitor, which fully prevented caspase-1 processing and IL-1 beta release from the infective cells. And all told, their data support their overarching hypothesis, and they were able to identify these regulatory circuits that repress flagellin and typhimurium by using a horizontally acquired regulatory two-component system that becomes highly active in this intracellular setting. They make the final argument that this evolved regulatory circuit limits inflammasome activation and dampens the host cell response from consuming the Salmonella typhimurium. So summing up, they walk us through a variety of experiments that demonstrate how the mammalian immune system imposes a selective pressure on intracellular bacteria to repress the expression of immune activating proteins, ultimately compromising host defenses. They end, though, with a very provocative notion, with antibiotic resistance on the rise and bacteria increasingly being linked to acute and chronic diseases, a thorough accounting of how bacterial immune evasion mechanisms may ultimately guide us in our thoughts of new paradigms in developing antimicrobial treatments is a must. So I thought this was a cool paper when I glanced at it, and I thought it was something that you all might enjoy.
Fortunately for Salmonella, downregulate flagella being intracellular doesn't matter, as we said, right? It's good. Otherwise, if you needed it, you wouldn't be able to do that.
No.
Yeah, I agree. This is a lovely addition to a large literature that shows bacteria shut off flagella, both for economy reasons, but also to avoid the innate immune system.
So some of this was known before. I mean, some of the important conclusions are not totally novel.
Yeah, the conceptual framework had been established, but they've provided some additional molecular detail and discussed it in an evolutionary framework. Right. But for example, Campylobacter jejuni and Helicobacter pylori, they've actually altered the architecture of their flagellin molecules so that it can't be recognized by toll-like receptors. Wow. So we know that there's a lot of selective pressure on the flagellin system, either shut it off or change the structure. The other interesting thing, and this gets a bit more sophisticated, but Brad Cookson's lab and others have been studying the populations of Salmonella in a mouse model, and they find that there is what they call a bistability wired into the genetic circuitry that controls whether or not they're making flagella, so that the majority of the population will be off, but a minority is on. And the idea is, and this is backed up by a lot of animal work, that a little bit of inflammation may actually benefit Salmonella pathogenesis in the GI tract. Now, if every single cell is alerting the immune system to its presence, then the infection is cleared. But what they've shown is that there's this bistability, so that a subpopulation does express flagellin, you get a bit of inflammation, and that seems to benefit the whole population. So it's a really interesting experimental model to understand host pathogen interactions, but also, as this group did, to understand evolution. And Michael, were you going to mention that they discussed this in context of a new, especially virulent strain of Salmonella typhimurium that's beginning to look more like typhi and cause systemic disease?
Okay. I wasn't going to do that simply for the lack of time. I knew I was going to run out of time if I do it. But now that you've mentioned it, go ahead and expand on it.
Right. The authors in their discussion point out that there is a particular clade of non-typhi Salmonella, so typhimurium, that usually stays in the GI tract, but it's now being found to cause life-threatening bloodstream infections. And when they go in and do careful analysis, they find that more invasive Salmonella typhimurium expresses less flagellin. So again, it's gotten better at hiding itself from this innate immune barrier and is able then to cause not only GI disease, but get into other tissues and cause terrible disease.
Let me ask you a question from far out. Couldn't something analogous happen with the O antigens external to the surface of gram negatives? That they are also a signal that here I am, a visiting card,
right,
or a neon light, like the authors say in an interview.
Yes. There is evidence that some pathogens will modify their LPS to make it less easily recognized.
Yeah.
And in fact, Bacteroides, one of our predominant gut microbes, has a relatively low level of annoyance to our immune system. Its O antigen systems aren't as bad as some of the others. So this selection has going on probably longer than humans can talk. So
I'd like to tell everyone more about the PhD student who led this project, Bushra Ilyas. She, I'm happy to share, said that TWiM has been fueling her passion for microbiology since she was an undergraduate at the University of Toronto. So while she was working in research labs, she would listen to these episodes and increase her knowledge base, but get inspiration. So she was an undergrad at University of Toronto, majored in molecular genetics and microbiology, and she did a thesis project with Will Navarre. So he also is a Salmonella pathogenesis scientist, and that's where she first became interested in pathogen evolution. So for her PhD at McMaster's, she joined Brian Coombs' lab, and we heard about their lovely work today. She said this research project extended earlier work from Suzanne Osborne and Brian Tunima, who had discovered that SSRB, this regulatory protein, was controlling other genes. She credits David Mulder for conceiving of the strategy to exploit the differences between typhimurium and bongari to understand the impact of regulation by SSRB. She also wanted to credit their collaborator, Victor Bustamante, and his trainees in Cuernavaca, Mexico, who did a lot of the molecular biology, including the gel shifts that identified FLHD as the binding site for SSRB. But reflecting back on the whole study, she says it's easier to forget about the frequent failures that accompany the few successes that actually go into the paper. So she wanted to share what for her has been the biggest lesson so far, so that junior researchers can follow this advice, and that is to tap the expertise of people around you. She said there were numerous experiments where she could have spent 2 or 3 weeks troubleshooting all by herself, but instead, thanks to the diverse expertise in the Coombs lab and also elsewhere, and his willingness to collaborate, she learned that by asking for assistance early on, you can really accelerate the project and learn a lot more. So she says it's an honor to share this success with all her co-authors, and she's taken pride in having a large part of her lab on the paper with her.
Oh, nice. Actually, this illustrates a principle. I think, especially when you start out, but even later in life, when an experiment doesn't work, you tend to say, "Oh, there must be something wrong that I did. Let me try again." That's not right. You should really think about what it is that caused the failure and consult with people who know better. She makes a very, very important point.
Yep.
Yeah. And science is very collaborative. We have this cartoon image of scientists just working solo in their laboratory with test tubes, but it's a really people-intensive business.
It must be nice when someone who has been inspired by TWiM gets their paper done on it, right?
She was really excited.
So you called her and told her we had picked her paper, and
then she fainted.
I actually emailed her.
Okay.
And then she responded right away and was really really generous and thoughtful with talking about her own path.
Oh, thank you, Michele.
Lovely story all around.
All right, that is TWiM 189. You probably listen on a phone or a tablet using an app. There are many apps, and whatever app you use, please subscribe. That way you get every episode, and it helps us. It helps get our subscription numbers up, so we would appreciate if you do that. If you really like what we do, consider supporting us financially. You can go to microbe.tv/contribute. We have a number of ways you can do that, including a subscription over at Patreon. And finally, we love getting your questions and comments. Didn't have time to get to them today, but next time, TWiM at microbe.tv. TWiM today has been Michele Swanson from the University of Michigan. Thank you, Michele.
Thank you.
Elio Schaechter, who's at Small Things Considered. Thanks, Elio.
My pleasure.
Michael Schmidt from the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
I'm Vincent Racaniello. You can find me at virology.ws. Thanks to ASM for their support. This episode of TWiM was edited by Ray Ortega. Music on TWiM is composed and performed by Ronald Jenkins. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.